Welcome again, everyone, to the 7th webinar of the Science of Aging series, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. For this series, we've lined up a number of webinars, all focused on the science of aging, being conducted by leading researchers from around the world in their relevant fields. This is Haley Culleton from Inside Scientific, and I am very pleased to be your host for today's event. Today's webinar is titled From Artificial to Real, What 24/7 Home Cage Monitoring Teaches Us in Preclinical Neurodegenerative Disease Models, and will feature Dr. Stefano Gaburro, Scientific Director for Technoplast, and Dr. Brun Ulfhake, Professor of Laboratory Medicine and Professor Emeritus in Anatomy at the Karolinska Institute. Throughout this presentation, Stefano and Brun will discuss continuous home cage monitoring and how moving behavioral testing to a setting that is familiar to the animals can decrease concerns of validity and animal welfare. Without any further delay, I am very pleased to welcome Stefano Gaburro to the floor. Stefano, please take it away whenever you're ready.
Okay, thank you, Haley, for the kind introduction. Good morning, afternoon, and evening to the audience, wherever you are. For those of you who don't know who we are, Technoplast was founded in 1949, and it made the first technological revolution in laboratory animals in 1962, when the first cage, plastic cage, came along. And then through revolutionary revolutions like washing, or 1994, the first IVC generation and laminar flow, we came to the latest solution, which is our home cage solution called the Digital Ventilated Cages, or DVC. But now you would ask yourself, why is the world leader solution in Vivaria move into a more digital or data company? Well, the reason being is that as a pioneer, we recognize a trend, which is already very prominent in Europe, but also getting very strong traction elsewhere, where welfare and in vivo science must go hand in hand, and you have to track your animals 24/7. Now, I would like to talk about two main aspects that are very prominent in today's research. Typically, when you do some behavior, for instance, or other type of experiments, you will move your animals from the home cage into, for instance, here, behavioral setup. But the recent research is actually moving back the animals and conduct experiment in the home cage. The reasons are several, but especially you want 24/7 recordings, and it allows to record the animals especially at night because the animals are more active there. You have unlimited observation time versus like a spotted type of experiment, 5 to 10 minutes. And if you think of aging experiment like Brun will talk later, or neurodegenerative diseases, where there is a slow disease progression, you have to leave the animals basically alone and do not stress that much. And that is why it's important to do the experiment in the home cage. The second aspect is that we have to distinguish between a real home cage or a temporary home cage solution. What real home cage solution, and this is something also we are preparing in the manuscript, which has been submitted to Frontiers in Neuroscience, is that when the animals are in there for the whole time of their lifetime, 99% kept. If you want to move the animals to do other type of tasks, like in this case, you can keep the animal for the time of the experiment, but then eventually you have to move them back in the home cage. So now, what we in the past decided to build is in the home rack, in the home cage, an infrastructure which is basically not even touching the animals, because it's a board that basically goes at the cage level, outside of the cage, and it's contactless. And this board is consisted of 12 electrodes. It's like basically pixels on a touch screen. This board generates small electromagnetic fields, as shown here, at 4 Hertz, so 4 times a second. And this can be basically modified by the cage, its content, or if there are mice in there. And we can basically convert this type of conversions, this raw data, this change in electromagnetic fields, into occurrences. And because we know where the electrodes are, we can calculate and derive several metrics, including animal locomotor index and others that I will present later. Now, if you want to have a quick overview of the research which has been done in the past using our technology, I wrote together with a friend, Wouter Levoika from University of Helsinki, a review which was sent to Frontiers in Behavioral Neuroscience, in which we basically summarize many applications which were performed with the DVC in the past. You can access it through the link or also download it from the resource panel in here. But if you don't want to spend that, I want just to give a brief overview of what has been done in the past. So first of all, the question is, do these electromagnetic fields do something to the spontaneous behavior of the animals or to the pathology? And we addressed that both in behavior and in pathology, and we could basically show that there is no alteration or substantial alteration that would modify either or. Additionally, because I told you that we can basically track the animal, if you have the animal single-housed, we can reconstruct the trajectory of the animals, and against video, which was considered the gold standard in behavior pharmacology, we could also reconstruct the speed and the distance traveled. And now I would like to show you two examples of recent publication which was achieved without technology. The first was published in Lab Animals very recently. And basically, these researchers are part of the International Mouse Consortium called IMPC. And they use group house animals to look in C57 Black 6 and Bouts mice, so inbred strain, and also outbred CD1 mice, and look for different spontaneous behavior in the home cage. And if you know what the goal of the IMPC basically is to generate several knockouts and study their loss of function in such mice. And therefore, it is imperative to understand what the basic physiology and behavioral repertoire are. What was found here in the slide, this is a heat map in which the color code represents red, it's the activity of the animals, which is very active, and blue is less active. What you see here is that you have the two inbred strain in the top and middle of the panel, and at the bottom are the CD1 mice. On the left side, you have the male. On the right side, you have the females. Basically, what you can really see at the first glance is that the CD1 mice in the night, which is where the red activity, the red spots are more clear, are far more active, irrespective of the gender, than the other animal model. And if you move here and you look the average activity during the day and during the night on the left side, you will also see that the basal activity in green in the lights on and in blue in the lights off is much more, they are more active. And on the right side, what you see is a 24/7 representation of the average activity of the different strains in male and females. And what you can also see is different peak of activity on the gray area, which is represented at night. And you can see that there is, according to the mouse strain, different activity, and you can derive several information out of that. What also researchers found was to assess something which is considered in laboratory animals not a stressful event, like the cage change. And upon cage change, there is a different response. So where you basically place the animals in the new cage according to the different strains, and also according to if they are male or females. And what you will see is that basically CD1 male mice have higher intensity of response as compared to the other inbred strains, but the duration of the activity is very similar to the others. So basically what researchers concluded is that the CD1 outbred mice have higher activity as inbred mice as a whole, and the response to cage change is more prominent in CD1 mice, the average activity, and the sex differences are most notable in CD1 mice. So now, for them was very important because if you have to generate the knockout from either, especially 57 black 6 mice, you can then study what the transgenic will do compared to the baseline. Now that we understand a bit of what the physiology basically, or behavioral repertoire, basal repertoire of mice is, you can also look at something more into detail and look into neurodegenerative disease, like in this case. So what here researchers look basically, they took a mouse model of amyotrophic lateral sclerosis, in short, ALS. And the researcher basically use SOD mice, which basically, typically in behavioral repertoire, they show the first symptoms of ALS at week 16. And they look also what can be basically shown in home cage behavior and compare to the typical behavioral repertoire, which is like the grid test that we'll present you later. They use pair house mice, male and female. And again, they look at the disease progression. What you can see here is another heat map of, at the top, you have the wild type, at the bottom, you have the transgenic animals. And what you can see in this white dashed line, which is basically emphasized on the right side, is that during the day, the normal animals have a lot of blue color activity, indicating that the animal is sleeping. And then every now and then potentially go and eat and then come back. So these are the red lines. And the long lines is when they do basically the cage change. If you compare to the SOD mice at the bottom, you see that the activity during the day is all over the place. You have blue, intermittent by red, and then blue again, indicating there is something going on with their potential sleep pattern. This is all nice and fine, but it's just a qualitative representation of what you can see, and you want to quantify it. That is why the researchers with our data scientists develop a new algorithm to basically assess this irregularity or chaos, if you wanted, the pattern of activity. And they develop a new metric called Regularity Disruption Index, in short, RDI, in which basically you assess the sample entropy, which has been used in the past to assess physiological time series like ECG, to assess cardiac issues. So what you see here on the left side, you have the males, wild type and transgenic. On the right side, you have the females, wild type and transgenic. And at week 16, as expected, you have a start of the increase of the RDI, indicating that this erratic activity becomes more and more prominent. It reaches maximum of at week 20. And then as soon as the animals start to become more immobile because they get sicker and sicker, these RDI start to decrease again. There is a little delay in the RDI increase in the female. When we move and compare with the grid test, which is basically placing the animals on a grid, put them upside down, and then you calculate how long it takes for the animals to fall basically on the ground and to lift the hind limbs, so measuring the muscle strength. You see on the top graph, I hope, that the animals at week 16, especially the males, start to lose this grip, and then the female is a bit delayed. And then the middle graph at the bottom, if you have on the left side the grid test and on the right side the RDI, as soon as the blue line as shown start to decrease, so the animals start to lose the muscle strength, the RDI start to increase, indicating that basically there is an overlay of the two things, but in the case of the RDI, you don't disturb the animals. I told you that the cage change can be stressful, and that is why we look if this is going to impact the RDI at all. And we basically can show that when there was a week or weekend cage change, there is no difference in the RDI in the control as compared to transgenic animals. So the RDI is going to increase irrespective of the cage change. And then when the researcher look for correlation and then try to see if there is any correlation between RDI and and activity, they basically show that in the main wild type at the top graph, there is a correlation as soon as the basically animals are in the pre-symptomatic phase, so they don't display any symptoms. But as soon as the week 16, 17, they becomes to be in this erratic activity, the basically correlation is lost between activity and RDI, as shown at the top and also at the bottom. The researcher also look for animal welfare, and they look at the body weight change, and basically they see that the control animals continuously increase the body weight, whereas at week 16 and more 17, the body weight of the transgenic animals start to basically turn around and then decrease, and then the RDI also is correlated to it. So overall, the RDI was used for this mouse model as a biomarker for identification of ALS, and there was also clear gender difference in the time development of the disease of all parameters. We didn't stop at that. We also tried to see whether in other neurodegenerative diseases like Alzheimer and Parkinson, there is also a change in the activity or RDI. And for instance, we could show, these are just preliminary data, that the locomotion in Parkinson model is very prominent at night, where you wouldn't look at generally to your animals, but during the day, where you can see there is no difference compared to the control animals. We were going to look also at the RDI and other parameters and see how the things develops. So I hope that I could show you that DVC helps scientists addressing more scientific questions regarding also data reproducibility, also and helps to identify clear pathological symptoms in advance, or at least help to do not stress the animals while you're doing that. And with that, I would like to thank you.
Thank you for inviting me, and hopefully my voice will carry me through this talk. And to some extent, my presentation will rely on what Stefano already presented to you. I don't need to dwell on why it's good to use home cage monitoring. I think Stefano made that message clear to you all. So if you move down to the bottom of that slide, we used it here to study rhythmicities, oscillations in spontaneous behavior inside the cage over a longer period of time in order to understand if there are oscillations that we induce by husbandry routines, experimental routines, or that are spontaneously existing among the animals. And furthermore, trying to get more detailed information of what is actually taking place under the curve of activations that Stefano showed could change in different experimental situations. So that is what I will show to you. And with rhythmicities, so they were defined in different types depending on whether or not they were generated by internal oscillators in the animals, and to what extent these depended on external triggers, or whether or not such oscillations were entirely dependent on external triggers. So you're familiar with some of them, like the circadian rhythm, of course, which is based on an internal oscillator, but that then trains to the daylight and then the 24 hours. We have slow circannual seasonal rhythmicities that are well defined in some wildlife species or small rodents, but have so far been said to not exist in our laboratory mice, especially not when we have them under controlled constant conditions. But we recently showed that these mice do show seasonal variation in behavior activity, and that this is something we have to think about in future experimental design. We have internal oscillators that we have to take into account too. These are the ones that generated the specific sleep patterns. These are those that guides us through the different phases of our life and so on. And then they are basically they can be impacted by external factors, but they are driven by internal oscillators. And then we have those that we actually introduce, and those are the ones that are triggered by external events. And as Stefano presented, and I will show here, one such reoccurring husbandry event is the cage change, which impact and will cause an oscillation in behaviors across in between these cage changes. There are other reoccurring experimental husbandry routines that may do the same. We use this way of obtaining undisrupted and data on spontaneous in-cage behaviors allows us to collect data over a longer period of time, and we can approach this data set with an unbiased data-driven approach. We have used different tools for this, and I will present how we have used them. We have used spectral analysis to understand clustering of activities with similar period. We have used functional analysis to understand, or to describe, I should say, recurring oscillations in behaviors. And we have used decomposition analysis, which is currently also used in studies of humans, where we characterize the activity and rest into bouts of activity and rest. And these have a period, and they have a duration, and so forth. And they can help you to understand a little bit better what is taking place in the cage. So this is the activity across 24 hours, as Stefano showed you. The black line here shows activity on the y-axis, and we have the hours of the day on the x-axis. And this is 10 cages of females, housed 5 to a cage, and each cage is indicated by a separate color, and the thicker black line is the average of the group. And we can see that they mainly rest during daytime, and the lights on, lights off occurs, triggers for increased nighttime activity. And the lower panel here shows the same recordings now from 8 cages of male mice of the same strain, B6, housed with the same density. And the pattern, the circadian pattern, is of course very similar, but as you can see, the level of activity during nighttime is different between sexes. Another way to look at this is to use the heat map, which also Stefano introduced to you. In this case, the panel to the right here is built up by 7 columns, 1 column for each day. And as you can see, set time, that's when lights go on. We have an increased activity. Deep blue is low level activities, and during daytime there's low level activity. And then at set time 12, when lights go off, activity increases. So this pattern is repeated every day. But you can see, if you look at this set of days in greater detail, that there is a variation from day to day. And that is a variation that we introduce by changing the cage. And I will come back to that later. Such a difference in activity, and like we see between day and night, and we see also in this circadian rhythmicity, slow oscillation in activity, of course correspond to differences in metabolic and energy production. So there is a very nice correlation between activity, and so this is 2 different cages, one in blue and one in red. We have day and night, lights off during the shaded area where we have high levels of activity and lower levels of activity when lights on. That's a calorimetry. So we have measured the energy expenditure during day and night, and we can see that there's a very nice correlation between energy expenditure and level of activity recorded in the DVC. So when we encounter large differences in activity level, this will correspond to a difference in energy expenditure and calorie consumption. Now, to understand a little bit better what is beneath the curve here of activity across the 24 hours, the circadian rhythm, we can do decomposition analysis. In this case, we have defined an activity as something that is sustained during at least 3 seconds. In humans, we use 1 minute as the minimum time That is far too long for mice, so we tried forth and back, and we arrived at the 3 seconds of sustained activity at the same level correspond to a bout. And then bout can be much longer than 3 seconds, of course. And here, just to make it simple, we have divided bouts into those with high activity in red, those low to medium activity in gray, and rest in black and white, with in black being then short bouts of rest, and those in white are long bouts of rest. And what we can see here, of course, is that the difference between males and females here is that there's much more red during night. So it's the high level activity that makes the difference between the sexes. But we can also see another difference that's not obvious for us from the shape of the curve. And that is that in female cages, the bouts of rest tend to be longer than they are in a cage with males housed at the same density. So this can help you to get details of what is happening when the curve shape changes. It's also good because it translates well to what's currently being done in humans. So we have same kind of parameters that we analyze. This is another example. It's average of 10 cages with 2 males housed to a cage, and the black line here is the circadian rhythmicity across 24 hours. In light gray, we have long bouts of sleep. In a little bit deeper gray, we have short bouts of sleep. In middle gray, we have low to medium level activity, and dark gray, that's high level activity. So this is the average. However, if we look at single cages, we can see that they are different. So the framed area here show these 2 cages, where the overall level of activity, if you count number of activations of electrodes, is similar. The amount of time spent in rest is similar. But we can see when we look in greater detail into the rest pattern that they are very different. So in the upper panel here, we have a cage where the resting bouts are short and only rarely are long. Whereas in the lower cage, it's as common that they rest in long bouts as they do resting in short bouts. So this can be used to analyze distortions in sleep patterns and rest patterns. So we can conclude that activity is context, strain, sex, and age dependent. And when we move from one day to another, or cages with the same strain, same age, same sex, there is a fairly close correlation between housing density and overall activity in the cage. So here we have used decrease in number of animals. We removed from 5 to 4 to 3 to 1, and then we increased the number of animals in B6, males and females, and as you can see, there is close to a linear correlation. And of course, the level of activity and housing density will impact the number of electrodes being used in any bout of activity. And they can also tell you a little bit about how the home cage is being used. And this is just one simple example where we show correlation between the average activation per second on the y-axis for a set of cages of female and male mice housed at different densities. And on the x-axis, we have the number of unique electrodes activated that second. And there is, as expected, a correlation with average activation per second, of course, and also housing density. Then we have this type 3 oscillation in behavior, which we introduce by husbandry routines and experimental routines. And this shows to the left here a 7 days or heat map covering 7 days, and with lights on and lights off. And we can see the response to lights on, and they go down to more resting light pattern, then lights off triggers their behavior. But then we can see, as the orange arrow points to the middle graph there, that once a week we do something, we change the cage. And there is a huge increase in activity during that day. If we then look at what happens the following days, then we can see that this spills over. So in fact, the response to lights on and the activity the following night is increased, and we slowly go back to the pre-cage change level. So that happens a little bit with different time constant, but 4 to 7 days. And using decomposition analysis, which are the two panels to the right here, from cage day before cage change to the left and day after cage change to the right. You can see that the major difference here and that the cage change imposes here is a dramatic increase in high level activity. That's the red, which has been encircled there by this yellow rectangle. But we can see one more thing. Even though the total amount of rest does not change very much, we can see that the amount of long bouts of rest decreases. So it selectively disrupt long periods of rest. And then we'll move over to this slow rhythmicity that we found from in our long recordings. So it's been speculated that this exists, and in wildlife animals it's certainly been evidence, but it's been claimed that this doesn't exist because we hold the animals under constant conditions, and the inbred animals are unable to generate this internal oscillation. So I will take you through this. The first slide here to the left is the same that we looked at when we looked at the husbandry routine-induced oscillation in behavior. And this was taken as 1 week out of a recording over 430 days, which we show here in the middle panel. So each day here is a thin stripe. And we can see here the dotted line at lights on, dotted line at lights off, generate the same kind of response across time. There is an overall decrease in activity, which comes with increasing age. But there is something more here. And if you look at the inverted triangles, you can see that there are periods reoccurring with higher overall activity. And this has a time constant of months. So what we did was that we plotted the average activity for a day against time then. And we obtained for these 8 cages that we maintained during the same period of time with the same density, same strain, same sex, this pattern that is shown here. And where you can see that the black line shows an oscillation in activity level over time. These were held in the same DVC during the same years, and thus we would expect, if there was any environmental trigger, that these peaks and lows will be synchronized across cages, but they are not. So they are driven by some internal oscillator, and the animals inside the cage, they all entrain to the same rhythm. Otherwise, we would not be able to record it. But across it doesn't communicate across cages, so there is no synchronization between cages. Now we shall look a little bit closer at one of the cages, framed there, A04. So this is a high-resolution spectrogram of activation oscillation, and what we can see is a gray pattern here around 0. 0 is average activity over time, and -2, +2, +4 is standard deviations away from the average. And we can see that we have a regular pattern of gray peaks, indicated here also with blue arrows. That's the cage change day. And in between there's lower activity. And this whole pattern of activity oscillation induced by the cages rests upon another oscillation, the red line here, which also shows peaks and highs, peaks and lows over time, but with a much, much longer period. We will take out now the framed area, framed blue rectangle here, and show it in greater detail. So this is a blow up of the same image. The blue single arrows there indicate day of cage change, and the days that follows are orange asterisks, and you can see that the activity peaks the day after cage change and then lowers until the next cage change. This oscillation in activity, and here we cannot see day and night. I mean, we could have done that too, so you could see actually the pattern of activity oscillation between day and night. But this pattern of oscillation now induced by the cage change rests upon this slow oscillation. And the period of this slow oscillation in this case was about 60 or 70 days. But we have now been through like 5 or 6 cohorts of cages. And the average period of this oscillation is 100 days. So it's very close to 3 months. And there's a magnitude here. So the magnitude, that's the amplitude between the peak and the low, which tells us a little bit how large is the difference between when they are at the low compared to when they are at a high. And that is roughly on average 65%. So it's a huge difference. And of course, this will have impact on the metabolism. So what we did, this is a recording covering 700 days. And you notice the gray bars here, which are the cage change induction or high activity, which rests upon the red line here, which is non-continuous, depending on that the cages were out of the DVC for a short period of time. And that shows the slow oscillation in activity with peaks and lows. And the green line is the feeding pattern. So we can see that there is a correlation between feeding and the slow pattern of activity oscillation. And this lags behind, so they are active, and then they compensate for that by eating more, and so on. So one way to analyze data sets, as I mentioned initially, is to use spectral analysis. And with Fourier transform, you can go through the data set, you can pick up periods that are very frequent, and this shows the number of cages that we have done put through spectral analysis, and we can see on the y-axis some very frequent periods like 12 and 24 hours. That's expected because that's generated by circadian rhythmicity. We have another peak at 7 days, that's the recurring cage change, and then we see that we have asynchronous peaks occurring after with a period longer than 2 months, and that's the slow oscillation in activity. This can be further moved to a 2-dimension. So in this case, we have the spectral analysis with the frequency on the y-axis, and on the x-axis we have time. So then we can see how this spectrum changes over time, and we could analyze the impact of experimental interventions or aging and so on the frequency and intensity of recurring activities, clustering of activities. What I will finally deal with is the use of decomposition analysis to define activity and rest in Bouts. So that is to understand a little bit better what's below the curve that we see here, which you're familiar with by now, and it's being used in a number of contexts. So in human studies with development, it's been lifespan studies, studies of depression in humans, and it's now also being introduced in high-throughput phenotyping of sleep pattern in mice. So we define here that a bout is a sustained activity that lasts at least 3 seconds or longer. And then you can define, depending on what kind of question you're asking and what kind of behaviors you're looking into, different levels of activity and rest. And this may help you to get more details out of the data from the DVC. And this panel now shows the cumulative frequency of bouts plotted against the duration on the x-axis for 10 cages of female mice housed 4-2 cage. So this is the overall pattern. It's kind of probability for each duration is kind of similar across the range here. But we can then decompose this into different levels of activity, which is the next panel show, where we have in blue rest, which is kind of similar to the overall pattern, and then in green we have low to medium level activity, and the vast majority, and in red we have high level activity, and we can see that the bouts of activities usually are shorter, especially the high level activity are short. Another thing that we can see for these 10 cages is that the variance, so the black line here is the average for all 10 cages, and the shaded area in gray to the left and in green, blue, and red in the upper panel to the right indicates the standard deviation. And as evident here, the standard deviation for high-level activity appears to be much larger than for rest and low to medium level activity. And this can, of course, then this is evident when we plot each cage, as the lower panel show here. Another way to do this is to use biplots, where we have 3 corners with rest in one corner, low to medium activity in the other corner, and high activity at the top. And we plot the number of cages with males in blue and females in gray during lights off, showing that females are more active and rest less during lights off. And then during lights on, and now the males are in light blue, the females stay in gray. We can see that they largely overlap, so the activity difference between sexes is minimal during daytime, but not during nighttime. And you can take this one step further and analyze life trajectories by this. So this is one example where the purple line in the middle shows the average daily activity over a couple of hundred, I think it's 63 weeks in one cage. And you can clearly see that there is an oscillation with a long period with peaks and lows. And you can then decompose this and see what is changing in terms of rest pattern, medium and high level of activity. So this is just an example. How you stratify activity and rest is up to the research you are conducting. I think I'm at the final slide now. So I would like to summarize that what I feel are the pros and cons for using this home cage monitoring system. In this case, I've been using the DVC system, and the basic system, the cons is that the basic system records only floor activity, and we cannot track individual animals when they are group housed, and the signal from the electrode may be distorted or impacted by other factors than the movement of the animals. But it's scalable and operates in real time or near real time. It can be fully automated, and it provides non-intrusive cumulative 24/7 data that you can collect over an extended period of time and get information that was previously not available. And it's very useful, as shown here and by Stefano, as a complement in animal welfare surveillance and behavioral phenotyping. And it's also possible to integrate or combine with other recording devices, so it's not limited to this. You can have a microphone in the roof and so on. I would like to emphasize, so there are a number of added values. This moving behavioral assessment to the natural habitat of the animals, it will be very important for us to get valid data, and maybe also this will help us to reduce the problems that we have with reducibility across laboratories. And we will have access to data that we didn't have access to previously, which will help us to get insights into the normal behavior, which were hidden to us, so to speak. And we will also be able to monitor what different husbandry and experimental procedures that we introduce, what kind of impact they have on the natural behavior of the animals. And I think one very important thing is that with a reasonable or a agreed curation standard, these data files can be built into set up libraries for strains, sex, ages, and so forth, allowing us to do data analysis that we have not been able to do previously, and to share data in a rather simple and straightforward way. I would like to mention that we are still in the very early phase of using home cage monitoring, so it's a very explorative field where we don't really agree, we don't have a gold standard, we don't have a common toolbox how to analyze the data. As evident from my presentation, we have tried a number of different approaches and so on. But there is a cost initiative now financed by the European Union, which will address these issues, and there's a link in the chat for those of you that are interested in following that initiative. Okay, thank you for listening. My final slide just is a disclaimer and acknowledgement. Thank you for listening. I'm prepared to take questions.
So, Brun, I'm going to actually bring this question up to your attention. I know you did touch on the number of mice housed in your cages a few times, ranging from 1 to 5. But the question here was, is there more than one mouse in a cage typically? So I don't know, would you mind just touching a little bit about on your multi-animal housing setups?
I think that single housing is something that we should refrain from as far as possible. So situation where we do studies or so on single housed animals is quite limited. So the common situation is that you have 2 or more animals in a cage.
Right. And then maybe a follow-up question to this was about single animal housing or isolation housing. Is it more stressful than a cage change or the electromagnetic field?
Sorry, you have to repeat that. What is more stressful, being single housed or?
So being single housed with the cage change or the electromagnetic field?
According to my own experience, because we have had them in and out, the electromagnetic field, we cannot record any impact on the animals by the electromagnetic field. The cage change, the thing is that when it comes at least to combination of males and females or females, so the natural habitat for mice is that they stay in groups. With males, it's a little bit different. Usually they stay with a number of females and in the wildlife, and they compete for dominance if another male show up. But when we hold them in a standard laboratory setting, we are usually able to group hold, to keep males also in groups.
Okay, wonderful.
Single housing should therefore be considered, especially for females, as stressful.
Okay. Stefano, the next one is for you. The heat maps that you showed, do they refer to single cages or to averaged out data with many cages? In other terms, can you assess the within strain variability?
Yeah, I think in the paper, the lab animals paper I showed before, there were heat maps for, there was an example of one cage, but there are also in the supplementary, all the heat maps for all the cages, but basically the one that are shown in the main figure showing basically that the CD1 mice are more active. There is one cage, and you can then assess the within strain qualitatively within strain variability. The cage change response was an average of 6 cages because there was only one line, but otherwise there was one heat map per cage.
Good. So one thing that's important to remember is that if any activity is not timed by an external trigger or is in synchronicity across cages, averaging is not good, because it will take away what is happening in the cage when the activity is not synchronized. So if you record, for example, a cage change or lights on, it's synchronized activity. So then I think you will see the same thing with averaged as with original one cage recordings. But when you look at, for example, the slow oscillations that we discovered, we would not have seen them if we had used average across cages, because they are not synchronized.
Okay. Another good question here. There was a mention of good translation of data to humans related to the decomposition analysis. Is there a scientific paper on this that you can share?
I can certainly do that, but I didn't prepare for it. So there are a number of papers that I could make available through the organizers here.
Sure.
Because I don't have it on top of my head. I don't have it, but yes.
That's great. Much of this data appears similar to David M. Lawson's work in the early 2000s using radiotelemetry. Are you familiar with this work?
Well, I think I can take this. I used to work with the radiotelemetry a lot before joining Techniplast, and I'm familiar with that work. Basically, it was work done in rats with different conditions, measuring blood pressure. I think here the difference mainly is that we don't implant anything in the animals, even if it's really nice that things are reproducible across rodents between mice and rats. And basically, in radiotelemetry, typically you will keep the animal single house, which is a kind of no-go, especially in Europe for research. And we can find the same findings, similar findings in mice without having to touch them, basically, and leave them in the home cage. This is what I can say.
Great. I think another thing that's important is I do appreciate telemetry because it provides a range of information that is rarely available, but it's not really scalable. You cannot run 2,000 cages with this, and so you have to choose the technique most appropriate for the questions you are asking. And I think that these systems should be regarded as complementary rather than competing.
Okay. And while we have the topic of rats and other rodents, is there a system available for rats, or can you use the DVC?
Not yet. We have it for mice. We have investigated for rats, but it's not yet ready. For now, at the moment, it's just for mice IVC cages from Techniplast.
Wonderful. We are a little bit over time, so thanks to those who have stuck around for this Q&A session. I'm going to ask one last question, but keep the questions coming in. We can always follow up. So last question today, the B6 has been well characterized. How translatable is that to other strains or stocks of mice?
I think that's a key thing that has been under investigation of some time. So there are a number of papers characterizing a multitude of strains and comparing them. The thing with the B6, of course, is that we know so much about it. We know so much about genetic variances in substrains of B6 and so on, and they were then favored as the strain to systematically do genetic manipulations on. So I would say that for example, when we obtain new observations, you have to expand it. You have to go to an upper strain. You have to move on further, like you do in industry. So you start with something that's quite well defined because it makes it easy for you. You control a lot of possible parameters that way. But then you have to be able to generalize. You have to use that pipeline of expansion. So you move on to an outbred strain, perhaps wild strains, different species, and so on.
I think if I can just add, also the tendency of the pharmaceutical companies is really to have a battery of different strains. Again, as Brun mentioned, mice, rats, and then larger species, and try to find the same findings according to the biological question which has been asked, and try to see whether it's reproducible or not. I know ideally, if you have mice that you get from the outside, you would like to have them, characterized and know that they have a similar pattern. I mean, it happened to me in the past that I was working with seizure, and all of a sudden I got a specific mouse from one provider, and all of a sudden didn't have a seizure anymore because they came from a specific barrier. And this is, you don't know from upfront, and this could impact your research.